I promise I shan't stand between you and the rest of your evening for too long. I am Rija. I'm a second-year PhD student with the Computational Cancer Genomics Research Lab at University College London Cancer Institute. And today I will tell you a bit about a project we've been working on the bootstrap inference of single-cell tumor phylogenies by integrating sequencing rate counts. So, as I'm sure all of you appreciate, recent single-cell DNA sequencing technologies allow us to accurately investigate heterogeneous populations of cells by enabling the parallel sequencing of thousands of individual cells. And a key application of this technology is in investigating cancer. So tumor progression involves the acquisition of genetic alterations, such as single nucleotide variants, or copy number alterations, whereby large contiguous regions of a genome are either gained or lost. And this happens via a complex evolutionary process, such that tumors end up consisting of genetically heterogeneous subpopulations of cancer cells. And by applying single-cell sequencing technologies, we ideally would have the resolution to follow SNVs harbored in single cells and use this data to reconstruct a tumor phylogenetic tree representing that evolutionary process. And this is important because it allows us to investigate mechanisms of evolution and has many clinical implications. And more specifically, the way we would do this is that given a conflict-free mutation matrix described by M cells and SNVs, where each cell is a row and each SNV is a column, and each value here denotes the presence or absence of that SNV in that cell. We can use this mutation matrix, conflict-free mutation matrix, to reconstruct the tumor phylogenetic tree under certain assumptions, the infinite site assumption that mutation can be gained once and only once during evolution and never lost. However, our data doesn't look like this idealized form of the matrix I just showed you. So you can imagine this ideal scenario on the left-hand side here, where we have this star variant present in one copy in our cell, and we apply single-cell sequencing technologies and observe some reads containing that variant, allowing us to make a true positive inference that yellow star variant is present in our cell. However, because of low coverage, we will very often see the situation where the star variant may be present or is present, but during sequencing, we do not observe any reads containing that variant, and then causing us to make a false negative inference that mutation or SNV rather is not present in our cell. And so what this ends up meaning is, instead of our mutation matrix looking like this idealized form on the left-hand side, it looks a bit more like what you're seeing on the right-hand side, which is a data set that is much more sparse and contains many false negatives. But that's not all. We also have the problem of false positives, where you consider the same situation where you have the star variant present, but not only is it not observed, instead what we see is a sequencing error, so this red triangle, and this might cause us to erroneously infer the presence of this triangle SNV, where in fact there was just a wild type. And so putting all of this together, our data actually now looks very different from this idealized matrix, because it's complicated by both the missing data as well as these false positives. And so inferring this true mutation matrix or the true phylogenetic tree from this type of data is actually a very complex task. But of course, this is not a new problem, and several existing phylogenetic methods are able to reconstruct the tumor phylogenetic trees and mutation matrices by integrating estimates of the uniform false positive and false negative error rates. So they attempt to correct these errors across this entire matrix. And the underlying assumption there is that these errors are uniformly distributed across this matrix. But this assumption is not true or does not hold. Instead, errors are actually non-uniformly distributed throughout the matrix. First, because error rates depend on sequencing coverage, which varies. And second, because you can have high error frequency genomic positions. So, for example, if we just consider one cell in our sequencing experiment, one type of genetic alteration that happens in cancer is copy number alterations. So within one cell, some regions may have either gained or lost large regions of the genome. So regions with higher copy number will give rise to more reads and vice versa, causing a fluctuation in the coverage along the genome and therefore the error rates within just one cell. But of course, in a sequencing experiment, we have several cells, and the sequencing coverage actually also varies between these cells or across these cells. Another type of genetic alteration that occurs in cancer is whole genome duplications, whereby the entire genome of a cell is doubled. So some cells will have double the amount of DNA compared to other cells. But even in the absence of this type of catastrophic event, just due to replication status, a G2 cell will have double the amount of DNA compared to the corresponding G1 cell. And this means that sequencing coverage and therefore error rates will also vary across cells in a sequencing experiment. And finally, there are some high error frequency genomic positions, and this can be due to alignment biases at those loci or PCR artifacts that were introduced during library preparation. And so, therefore, this means that errors are actually completely non-uniformly distributed across our mutation matrix. And so the key idea that we had is that in order to accurately correct these errors and infer the true matrix, we must integrate the actual raw sequencing data, so the variant reads matrix and the total reads matrix, into our inference. So to do this, we introduce a novel algorithm, Phantom, for the bootstrap inference of phylogenies by integrating sequencing read counts. Broadly, I'll explain the key features of our algorithm in a few steps. First, what we do is we take the variant and total reads matrices as input and resample them to create an instance of the input of equal size for which we solve the mutation matrix and then the tree. And the way we do this is by first reordering these resample input matrices and then carrying out multiple hypothesis testing in order to infer the corrected genotype matrix. And this approach, or this idea, is an extension of a fast heuristic introduced previously in the Huntress method, where they showed that given an input binary mutation matrix with errors, we can infer a corrected error-free genotype matrix using a fast heuristic that broadly consists of two steps. First, what we do is we actually reorder our matrix in the such that mutations that are likely to have occurred early during evolution are ordered first. So we order it in terms of most observations. And then given this ordered matrix, we recursively infer, using a top-down approach, the corrected matrix such that where each column, we use it to identify other mutations that occurred in its subtree and use this to correct errors. And in Phantom, we extend this idea probabilistically to take read counts into account such that we have more power to distinguish errors from truly observed genotypes and more accurately correct these. And so just to give you a flavor of the type of reasoning that Phantom would be doing under the hood is that for each SNV in each cell, we can carry out hypothesis testing. So we can test, for instance, if we have enough evidence to reject or not reject whether the number of variant reads that we observe might have been due to a sequencing error. And analogously, we can also test if we observe enough reads to reject or not reject whether that variant is truly present. But so far, we've created one instance of the resample input and solved for one mutation matrix and reconstructed one tree. But we don't just do this once. Instead, we bootstrap the inference by repeating step one multiple times to obtain this distribution of plausible topologies, each of which corresponds to one instance of the resampled input matrices. And this allows us to mitigate the impact of high error frequency genomic loci, as well as overcoming the limitations stemming from subsampling SNVs and reconstructing a phylogenetic tree just once. And finally, given this pool of plausible topologies, we determine the final tree by combining the most prevalent mutually consistent clades present across all trees. So we tested the performance of our method by extending the TracerX tumor evolution simulation framework to the single cell context. So here's an example of a simulated tumor phylogenetic tree, the corresponding true matrix, simulated sequencing data, as well as an example of the inferred mutation matrix. We ran both Phantom as well as existing methods and found that Phantom here in red improves the accuracy of single-cell phylogenetic reconstruction compared to existing methods, both in terms of the accuracy of the topology as well as the ancestral mutation accuracy. And in fact, this improvement in performance extends to different single-cell sequencing settings where we vary the coverage and the relative numbers of cells and mutations that correspond to different real-life single-cell sequencing technologies. But is this improvement in accuracy actually biologically important? To answer this question, we applied our method to a previously published dataset by Tyler Funnell and colleagues of single-cell DNA sequencing applied to a high-grade ovarian serous adenocarcinoma cell line comprising about 1,000 cells and 9,000 SNVs. So first, I'd like to direct your attention to this heat map over here. What you're looking at is cells in every row and looking at the copy number state across the genome. So here, even without reconstructing the phylogeny, without looking at the SNVs, just considering copy numbers, we can recover these three clear copy number clones, the blue, the green, and the pink. And when we applied our method Phantom to reconstruct the phylogeny using just SNVs, we were able to recover this clear branching structure corresponding to those three copy number clones. So here, every leaf is colored by the copy number clone that cell originally belonged to. And here, our method was the only one that successfully ran within the time limit using the entire input data set. We also ran the other methods using a subsampled subset of the mutations as input. And we found here that these other methods failed to recover the branching structure consistent with copy number evolution. And the reason it might be important to accurately be able to do so is that without doing that, you might miss the evolutionary history of key genes. So for example, we found an oncogenic driver mutation in most of the cells in the pink branch that would otherwise have been missed had we not accurately reconstructed this branch. And lastly, because we construct multiple trees, when we run Phantom, we can estimate the confidence we have in each edge of our inference. And finally and briefly, we have also started applying our method to a large single-cell patient cohort of 31 patients, also published by Funnell and colleagues. And here we found that Phantoms, so here this data is colored by patient. So here we found that Phantom's inference of phylogenetic trees being consistent with copy number evolution was a result that seemed to generalize across patients. So to conclude, we introduce a novel method Phantom for reconstructing single-cell tumor phylogenies by integrating sequencing read counts. We demonstrate the improved accuracy of our method across a range of simulation settings corresponding to real-life sequencing technologies. And we show that our method infers trees more consistent with larger genetic alterations compared to other methods. I'd like to thank the organizers for the opportunity to present, my supervisor, Simone Zaccaria, the rest of the CCG lab, our funders, patients and their families. I'm happy to take any questions.
Yes, we have time for questions. Nobody? Do we have any online? A question there. Thank you.
Hey, thanks for the very interesting talk. I just have one question. So you did an explicit comparison between clonal evolution identified via copy number and via SNVs, Have you looked at all at the direct integration of the modeling process with those two modalities rather than the comparison post hoc?
So you mean as in trying to reconstruct using copy?
Using both copy number and SNVs, yeah.
Yes, so we haven't looked into that formally yet. That would be a very interesting next sort of step on the horizon. What I would anticipate are the slight complications with that is that just the models, the generative models for copy number evolution are very different and quite challenging to put together. And actually, my colleague will talk about copy number evolution tomorrow. But I think it would be interesting and cool to be able to do that, but it would require a huge change in the thinking of how we do it.
Okay, thank you very much.
Thank you so much.